So yeah, this is where I grew up, and I negotiated to have a lab here, and then I'll have King Kong tear it all down.
You know, the last owner ended up very badly, so.
Yeah, so actually I moved to Munich, which is pretty close to this. I think I'll come from a slightly different perspective, ending up with similar problems than you have, but trying to walk you through the motivations or the path, how I came up ending in such a conference. So I think basically one can maybe say that my interest starts if you, for example, go to the Leipzig Zoo, which you definitely should if you're there, because they have a great great ape facility with all the great apes present, like the orang, gorillas, the bonobos, and the common chimpanzee. And I think what many people feel, including me, is that one is fascinated by, on the one side, that they're pretty different, right? I mean, they have definitely more hair than I do, and slightly larger ears, etc. But then, on the other side, they're also so damn similar, right? So they like some things I also like. So between this being similar and different, actually, you can understand a lot of the literature, because some stress the one and others stress the others, and that gives different viewpoints. Be it as it may, this is what would be interesting for me. And one of the first questions, this is an outdated slide, but I didn't dare to update it. So the first question is really about relationships. That's, I guess, not only for this case, but for many others. So basically, who's the closest? And historically, that's interesting. There was a recent book that goes on the history of great apes. So the first great apes arrived end of 17th century, and the first description are from sailors in Africa. And actually, funnily enough, one of this description from the sailor says that they think probably the chimpanzees or gorillas that were jumping around in the forest are aborigines which do not talk because they're afraid that they're going to be put to work. So, but back then already, 1699, Edward Tyson clearly realized that this orangutan, as he called it, but it's actually a chimp, was in many anatomical features more similar to humans than to a monkey. And then after Darwin, was kind of clear that either the chimp or the gorilla is the closest relative of humans, and then it took almost another 150 years to resolve that, because you could assess relationships by DNA sequences, right? Which is took a while to persuade classic zoology that all of their phylogenetic anatomical measures would be just not as good as sequencing DNA to determine phylogeny. But I think now this is kind of, there's no doubt about that using DNA to determine phylogeny is kind of the way to go. Why? Because you inherit your DNA, obviously, from your parents, so you have vertical transmission. You have also what Simon nicely pointed out, recombination. So you do not inherit a whole chromosome from mother or father, but actually a mixture of that, which is exactly the kind of mixing of genetic material that is used if you do an association study. Or that is also the case, Dorothy pointed out that if you have a brother or sister, you're on average 50% related, but that's because 50% goes back one generation, right, which is essentially identical, and the other 50% are just as related as your grandparents were, or two normal people from the population, right? So it's pretty clear that during this process, if mutations occur, you get information about a relationship. So if you, for example, now sequence the chromosome here, this part, from one person or the other, you might find a sequence difference. And why is there a sequence difference? Because back in time, right, there was one inheritance where there was a copying mistake, in this case from a C to T, so we end up at one point now where one individual, or actually one chromosome more precisely, has a disposition a T and the other one has a C. And you can see that if this process is constant across time, that it just measures time, right? And also what is important, it measures time especially well if this C to T change does have no effect on the phenotype, right? So evolutionary if it's neutral, which most of the changes in the genome actually have. Okay, so if you spin that further, one easy thing is that if you have 3 chromosomes or individuals, you can actually infer by the similarity of these 3 sequences a relationship. Of course, you need more than just this one position, but the principle is kind of clear, right? If these 2 individuals have a C at that position, but that a T, then they are on average closer related together than this. And if you acquire enough data, you can draw these trees, okay? It's also given the rate of mutations, it's often also pretty easy to infer which was the ancestral state, right? So it's very unlikely that that there is 2 changes from a T to C on both of these branches compared to this, right? So the more parsimonious explanation is because mutation rates are low if you compare humans or closest relatives, usually very good. Okay, so if you keep in mind that the changes basically represent time, because they occur proportionally to the amount of generations the sequences have passed through, then if you sequence random pieces of a human and a chimpanzee genome, so the same orthologous pieces, you get this number, which is pretty famous, right? That 1.2% of the sequence is different. And of course, it depends what you count. So you count, in this case, point substitutions, right? One base is a C in the one and a T in the other. You do not count that there are 300 bases in addition, right? That's a different way of counting. So if you count substitutions, you get this number, which is often confused that 1% of the genes is different, right? That's sequence, and 1.2% of the sequence is different. If you do the same thing for chimps and gorillas and humans, you get these numbers. And essentially, you can fill in this matrix of all these species and these average differences. And it turns out really nicely that these are the genome-wide averages of point substitution differences, making it very clear that on average, this is the phylogenetic relationship of humans, right? There's no doubt about it. And because 1.2% is the difference between a human and a chimp or a human and a bonobo, then of course both of them have the same time behind them, so that's basically this time point of the common ancestor is 0.6% substitutions ago. For me as a molecular biologist, that's a total fine time frame, and I don't care really so much about the real time that relates to. But of course, if you know how much percent you get per million years, you can transfer that into real time. Interestingly, recently there has been a new debate actually how you transfer that. Originally, that corresponded for a fossil calibration point to about 6 million years. But now they have measured with next generation sequencing actually the amount of mutations you have per generation in current humans, and that gets it more up to 10 million years. So it's not really resolved yet how the 1.2% translate into real time. And you have a range of 5 to 10 million years, which is not when the species split one needs to consider, right? That's the average DNA tree. Of course, that's kind of the average time back to a common ancestor. Of course, this is longer ago than the species split, right? Okay. Anyway, so you have this average DNA tree, and of course, so if substitutions represent time, then you can do the same thing with 2 humans, and if you measure that, you get approximately a little less than a tenth of that, right? So you can remember 1% and 0.1%. That's kind of fine. Which means, again, on average, across the whole genome, if you compare 2 chromosomes or 2 humans, and interestingly, if you do that across the world, this number is not so different, which is a long finding. So if you go further away geographically for the comparisons, the number does increase, but it does not increase that much. And of course, again, that you can translate into time depending on the clock you have calibrated. And of course, there are some regions of the genome where one has pretty close ancestors, and others where the ancestors are pretty far away because of this recombination issue, right? They're just independent due to the recombination. For one piece of the chromosome, we can be pretty close relatives, and for another piece, we can be very distant relatives. Why is this number so similar? Because, so if that's on average 500,000 years, and we know from genetic and also archaeological evidence that we really migrated out, say, at least not longer ago than 100,000 years from Africa, that means that most of our genetic relationship falls back into a time when we all were in Africa, right? That's the way it is. So one can view basically the humans across the planet as Africans, with some of them living in exile recently, if you want, right? I think that's a proper description. Why did that come a surprise to some of them? Because there are pretty big differences in skin color and actually also hair structure across the world, which correlate pretty well with UV light. And maybe humans tuned psychologically to find differences between groups, overstress the differences across the continents probably, so that's why they're surprised, meaning that the average genetic relationship does not correlate well with skin color. And you see that's Schwarzenegger, a little bit colored, with a little bit of different hair. And as you all know, he's Austrian. Yeah. Okay, so if you go across the world now and look at different populations or individuals that come from different places, that's about kind of the relationship you have across humans together with our closest relatives that still live, and also with our closest relatives that are extinct. My former boss, Svante Pääbo, was pretty famous for sequencing Neanderthals and these other species, Denisovans. So that's kind of the average relationship. But as I said, right, that's just the average over the whole genome. Every single locus across the genome can be very different, right? So averages, distributions, right? That's a general thing, but one forgets it. So for any particular DNA tree, you can have a relationship where the African is closer to the French, and then closer to the Chinese, and then the Neanderthal is actually closer to all of these three compared to the reference genome, etc. Right? You can get all of these different combinations if you look at the whole distributions. The trick is if you have now a gene, right, that goes together with skin color, with light skin color, then this tree will of course look different, because then this tree will tend to cluster with light colored people, right? And that's also something that you can use to find these genes.
What's the reference genome?
What?
What is the reference genome?
So that's just technically the thing that you find in the Human Genome Browser, but it's basically in European.
Isn't it basically Craig Venter?
Yeah, for the Craig Venter genome, it's Craig Venter. But for the other one, it was a distribution of a few unknown donors from the US. Okay, so that's actually interesting because very few regions will actually show local clustering, and in this way you can find maybe genes that were selected during the colonization of humans, and a couple of people do that, but that's not kind of the average representatives. That's what I want to say here. You could also look for genes, actually, and that will become relevant maybe later on, where actually all humans are pretty closely related in comparison to overall mutations, or maybe also in comparison to the Neanderthal as an outgroup. That would mean that that here's a gene that spread pretty fast recently, right? So everyone is pretty closely related for this particular piece of the genome, and that can be a statistical signature of selection, right? Basically, if the region is very young and everybody has it, that can indicate that it's spread because of positive selection. And these kind of signals population geneticists try to use in order to infer function, and I come to that a bit later, okay? All right, it's not only that we have these average estimates, we have it actually a full catalog of these differences because we sequence the chimpanzee genome. And if you scale up these 1.2% to the whole genome, that adds up to 35 million nucleotide substitutions, which 1.2% doesn't sound so much, 35 million does. Yeah. These are, of course, as Simon already nicely introduced, not the only mutations you have, right? These are the mutations you use in order to measure time because they behave very nicely in order to measure time. But there are, of course, also other mutations like these chromosomal rearrangement, insertions, deletions of DNA, and most of them being very small, right? 1 or 2 bases that are added or removed. Some of them can be pretty big, like millions of bases. And as events, there are about 5 million of them, right? So overall, there are less events, but they can, of course, affect partly very large regions. Yeah.
How many? What's the N for the chimpanzee genome? Supposedly, it's much smaller than the human one. Right?
The what?
The N. How many samples has been?
So recently, there have been sequenced more chimpanzee genomes. If you look at the tree, really, I mean, you could add chimps here, right? Overall, chimps have more differences between each other, depending also on the geographical structure, than humans, which would mean that on the long term, there were more chimps than humans. Yeah. But on average, right, I mean, you take one chromosome here and one chromosome here, because it's the same time, right? It doesn't; these numbers would not change depending on how many chimps you sequence here, right? Okay. So actually, technically, it's important to know these large insertions of deletions of DNA are technically pretty hard to detect because you sequence DNA in small stretches. And if you have 2 stretches that are very similar, it's actually pretty hard to say, are there really 2 pieces or is there just one? Which means that one has an estimate of this number, but concretely the catalog of these differences, especially the long insertions and deletions, is not as good as the catalog of these ones. Anyway. And then, of course, not only insertions, deletions of DNA, but our chromosome 2 is a fusion of 2 ancestral chromosomes, so you have also other kind of changes, right? To break that down to proteins, and let's say a few words more a bit later, these are about 60,000 amino acid differences, okay? So if you then, and we have by now, have also an outgroup, you can, by parsimony, infer which of these differences happened on the chimp lineage and on the human lineage, right? So if the orang and the rhesus has a C, and the humans have an A, and the chimp has also a C, then it's very likely that actually on this lineage there was a C to A change, right? And that works fairly well. So you had 35 million nucleotide substitutions, 5 million insertions of deletions between humans and chimps. You divide that by 2, that comes up to 20 millions, or a little bit less, a little bit more than that. That happened on this lineage, and then, given the variation you find in humans, about 80% of these differences are expected to be fixed, right? You have them; they happened before the most recent common ancestor of all modern humans. And then we have also the Neanderthal and the Denisovan genome, so for some we can actually distinguish whether they happened on this small lineage. But this lineage is fairly small. So, and yeah, 30,000 amino acid changes. So that's basically these are the genetic changes that make us human, right? So I could read off now the list for the rest of the talk, and you could regard that as an answer. Maybe not a very satisfactory one, at least not for me either. Because what you want is really, you want to have a genotype-phenotype map of these changes, right? That's what you really want. You want to know which genetic changes or which group of genetic changes actually correspond to phenotypic differences that happened also on this lineage, right? So genotype-phenotype associations are kind of a major problem of genetics, as you have heard several times now. And it's, of course, much easier to find them out if you have still active recombination, right? If you have a breeding population, you mix through all the different genetic variants, and you can do, as you have heard in the last talk, association studies that try to associate particular variants with particular phenotypes, right? That's also already pretty difficult, especially for these small effect sizes, but at least you can do it. For these fixed changes, you can cross humans and chimpanzees, at least. Maybe even it's possible, but I guess it's not really, I wouldn't suggest that as a project. So as a proper geneticist, you would actually say the system sucks, right? I mean, it totally does, because, yeah, you can't do genetics if you can't do crossing, right? I mean, that's ridiculous almost. But if you kind of have a PhD in the Max Planck Institute for Evolutionary Anthropology, it's actually not easy to switch systems, right? Because that's actually the system you're interested in per definition. What you also can't do is, right, there would be kind of a, you could experimentally mix up genotypes. But that's, of course, also not possible, right? We could neither change genetically chimpanzees to a human state in order to find out whether a particular genetic change actually does something to a phenotype. So the two main things you would have in a genetic model organisms are not available. And you either move to other species or you see what you can do. So that also basically means that for the fixed genetic changes that should contribute to the fixed phenotypic differences, we need to add information, right? There's no way out. We need to add information, and this information can come from several sources. And I try to lead you through these sources because basically all the human genetic, all the arguments for particular genotype-phenotype associations during human evolution use these three kind of sources in some way or the other. So the first source is maybe the most obvious and the most important one actually, is that not all changes on the genome are equal. So the prior that there are actually have an effect is not equal, because there are large parts of the genome where we think there's actually no function associated with that. So that comes down basically to the genome annotation. So if you have a G that you classified as having occurred on the human lineage, the question is, in what genomic context is it, right? You basically want to know where are the genes and where are the regions of the DNA that regulate your genes, right? That is the functional context of the genome, right? That's the source code of the genome. And you have introns, as Simon nicely pointed out, but basically it comes down to that you have a structural part that for the proper classic genes encodes a protein that then can regulate back to the DNA that carries the information when and where this protein is made, right? It's not necessarily, you don't have to make a protein of an RNA. The RNA can work by itself, right? MicroRNAs, long non-coding RNAs are examples. They are just beginning to be understood, but the principle is still the same, right? It doesn't really matter if you translate a protein or not, but this circuit is the one that you want, right? The protein coding gene, we have a pretty good idea. There are 20,000, which is true for essentially all mammals. So this is kind of the basic set of proteins. And if you calculate how much that makes, right, you come up with 1% of the whole genome. No.
Maybe I wasn't so strict.
I mean, it depends what you really count. If it's the mRNA, right, that also has the 5' and the 3' UTR, then you come about 1.5%. Yeah. If it's just about what makes protein.
You unlocked that extra 1%.
Yeah. Just ask Scarlett anyway.
So if you just ask about which part of the genome actually becomes an amino acid, then this is the 1%. And interpretation-wide, this is important because the the easiest interpretation if a change changes an encoded amino acid, right? Whereas all the other changes are much harder to interpret. Okay, so we have 1% of the genome is coding, and that will not change, right? This is, by all we know, this will not change. It will change for individual genes, maybe here and there, but overall, this is very stable. There's a big question, kind of what proportion of the gene outside these coding regions is actually functional. And a large part must be the DNA that regulates these genes, right? And there's also a question how many non-coding RNAs are actually functionally important. And this is not really clear, but say it's probably less than 1%, very likely, by all we know now. So the regulatory DNA, whereas it's pretty straightforward to infer where you have mRNAs, because you can sequence these mRNAs and know where they are in the genome, it's pretty hard to define the regulatory DNAs. Essentially, we have very little clue where they are, right? In order to get an idea how much of there is, the best way to define that, what is functional, is by looking at conservation, which basically means if you have parts of the genome that change less than you expect across species or within humans, then you infer that this region is constrained, and if you calculate how much of the genome that affects, then you can get some idea how much of that might be, right? And that people have done. So that's also possible by, if you add, say, all mammals, all vertebrates, genome sequences, you have particular regions where you have few mutations across these species. That includes almost always the exons, but also includes parts that are outside of the protein coding regions and are, in most cases, thought to be regulatory regions, okay? 5% of that are conserved within mammals, right? So if you subtract the 1% or the 1.5% for the mRNAs, that makes 3.5% of regulatory regions. More recent estimates from human variation say that there should be an additional 4%. In the very recent paper, that's just a week old or so, is, I guess, the best estimate currently, and this is about 8.2%. So function is, of course, also a quantitative issue, right? Especially conservation is if you change a particular site, you can be either immediately dead, right? That's pretty functional, or you can produce on average 0.001% less offspring, which is maybe just a little functional, right? So function is not an absolute term. It's really a relative term. And this number might increase if you maybe get more sensitive how much function we still can detect, but eventually you might call something not so functional if it has an effect to reduce your offspring by 0.001%, okay? But I mean, there's been a big debate because the ENCODE consortium considered everything to be functional that is bound by a protein, and they came up with this number of 80%, which is really totally stupid. I mean, really seriously, that makes no sense whatsoever. Yeah, okay. So the problem is that we have this number, but we have not really a good idea which regulatory regions are important in which cellular context, right? So although we have this number, we can't localize it that well still. That's worked on, but it's still a major issue, right? Yeah, so that's summarizing that. So that's kind of good news, right? I mean, there are regions of the genome that are much more likely to have a functional impact than others, although, of course, I cannot be 100% sure. There is certainly information coming from this annotation. If I now consider a unit, like a gene or a regulatory element for a particular gene, I can, of course, have additional functional information. For example, I know the biochemical activity, right? I can predict that this is a hydroxylase, or I actually know the protein structure. And the problem, of course, is that it's pretty hard to infer from such an information which phenotype is affected, right? That's just pretty difficult. What is also very informative, and one has information, is where and when a particular gene or even regulatory element is active, right? The problem is, especially for 20,000 genes, they are active very often all over the place, right? I mean, you just have 20,000 genes. Very few of them are highly specific. So you will find very few genes that would be active in Broca's area, if any, that are exclusively active in Broca's area. I think there's probably none. So although it's of course important to know where and when a gene or an element is expressed, because that constrains where it could actually do something, in reality that does not constrain the interpretation very much, because genes are expressed in many different places in many contexts. What is also an informative annotation you very often have are what happens if a mouse or a human has a null allele, right? If the gene is destroyed. This is the most important information or the most functional information you can get. And that's, of course, very important. And for example, there are consortia that systematically knock out all the protein coding genes in the mouse. So it's not so long until you will have for each gene kind of a database ontology that describes what happens with a mouse when this gene is not present, right? In many cases, that will be mouse is dead. And that's, of course, then unclear how you actually relate this type of allele, a null allele, no function, to maybe an amino acid change that happened during human evolution, right? This is not totally clear how that would work. So that's important to consider, that null alleles are not the same as an amino acid change. And also, of course, if you, for example, interested into brain size, and you find a bunch of genes that reduce brain size if they are destroyed, there are probably many ways to break a system, but not so many ways to kind of change a working system, of course, right? So that one should just consider. It's also unclear how often a phenotype in the mouse will actually turn out differently in the human genetic background. Another type of information we have are linked variants associated with human disease, right? Association studies that have been described, and this information gets more and more. And that's also, I mean, I'm being a bit overcritical here because I said the negative points. It's clear that this information is all very important, but one should keep in mind that, of course, the variants that we associate with phenotypes are often disease phenotypes. And so we know quite a bit about diabetes, but actually that's not a trait that we might be interested in during human evolution. But on the positive side, I think if association studies get cheaper and cheaper, we might actually think of doing association studies, particular for human changes in phenotypes that we're interested in, right? So, yeah, currently it's mainly diseases we look. And then also what is, I mean, you link variants to a particular haplotype or a region, and very often not to a particular variant, right? So you know in this region there's an association, but you do not really know often whether this is a regulatory variant or a protein change and whether it's up or down regulation, etc., right? There are limited information. Yeah. Nevertheless, I mean, all this integrates into your knowledge or your information you might add to a particular genetic change you want to interpret. And that's actually very similar to some extent to the problem that the clinicians have if they now sequence genomes in patients and they want to interpret which of the mutations actually matter. They have to do a very similar thing. So you can, you can view that as a kind of Bayesian approach to, you have a genetic change and then you take available information and try to interpret that. And the hope is, of course, that the more we know about biology, the better we can do that. So it's not only about, so that's of course only one side. The other side is that I have to define which phenotypic changes I actually want to explain, right? Obviously, I mean, also that's true for diseases. I need to measure my phenotype pretty precisely. And that's, of course, there's much less known than we would wish, which phenotypes actually do differ between humans and chimps. Not to talk about extinct species where we have really very little clue what the phenotype was except for the bones. Some phenotypes are pretty interesting, but the numbers that they have been investigated are pretty low. There are claims, for example, that cancer susceptibility in great apes is much lower than in humans. Also, Alzheimer's susceptibility has been claimed to be lower, but in my take, just the number that have been looked at are not high enough to be really sure. But I think, in principle, and Ajit Varki is really fighting for that, it would be very helpful also for the chimps to get better phenotype descriptions. And of course, if you have a phenotype that is very loosely defined, that, like with association studies, that doesn't help you, right? If you just say, oh, I know we are more intelligent than the chimps, that's probably not very precise and does not really help you to link to any genetic traits. And some phenotypes, for example, expression differences in the brain, can be measured quite well, but then it's of course not so easy to interpret what functional consequences that has on later levels. There's a third type of evidence that you can use, and that's something that comes from statistical analysis of comparisons between genomes and within humans. And they all want to infer essentially positive selection, meaning kind of changes that had a selective advantage. Either very recently, so I sketched this scheme where you have a region in the human genome where every human is very closely related that would indicate that this is a region that grew very rapidly in the last 100,000 years and might have been driven by a selective sweep. But that is confined to positive selection that happened pretty recently, right? So the statistical signal that a genomic region is younger than the average does not last very long, as you can imagine, right? And of course, also it's a statistical signal with not terribly much power because there's huge variation already what you expect, and if you need to be really on the extreme side to detect that. But nevertheless, that's one way you could detect positive selection. And the other one, which can be detect selection on the whole region, is essentially if you have on a particular gene or protein repeated selection, right? You change it again and again, then you can get more mutations in this protein than you would expect. And if you have more changes than you would expect, you can interpret that as positive selection. Imagine on average you have 2 amino acid changes for a given protein of a given length, and all of a sudden you have 20, you say, ooh, okay, that's much more than I would expect, so that's actually very likely that you have positive selection. And this is this measure that is often used, it's called KAKS, so you need to normalize kind of the changes you see in a protein to the mutations that you expect, and you do that by comparing the substitutions that change the protein compared to the ones that do not do that. Anyway, what is important here is this is fairly powerful, but confined to very recent selective events. This can cover the whole lineage, but you need strong selection. So the biggest issue is that that it's a very nice system, right? I mean, you just need genome information and the statistical analysis, but it has a high false negative rate, right? I mean, it's totally clear. You do not have endless power. Also, the false positive rate is actually pretty difficult to control, but that's something you can do, of course, better, right, by increasing models and data, etc. Very often, if you just go to the databases and screen for positive selection, what you actually find, the artifacts, right, where you have not aligned the human and the chimp genome correctly, because you're basically asking for artifacts if you look for outliers. It's inherent in this thing. But I think that can be controlled, but the false negative rate doesn't go away, of course. And then it's also a question of kind of what effect size you detect. For example, in Drosophila, one thinks that between 2 species, over 90% of all the amino acid changes are positively selected. Oops. That doesn't all of a sudden help me much, right? If any change is positively selected, that doesn't give me much information, right? So it's again, as always with statistics, I guess, if your power increases, it's the matter of effect size that you talk about, right? So positive selection is not a 0 one thing, but it's actually a question about how strong it is and actually how the distribution of positive selection looks like. And also what is important to note, not all positively selected changes must be linked to adaptations. So imagine you go to a period of evolution in a species where you have small population sizes, and if you have small population, shit happens. So maybe you have a deleterious allele that fixes in the population, and then you grow again and have more power to have selection, and then you compensate for that thing that got messed up. So you can compensate, and that will lead to positive selection, right? So you can try. In other words, you can get positive selection just for stabilizing phenotypes. So positive selection is not really equivalent to adaptations, right? Because what we want to know the genetic changes that make our human niche really unique, and not so much whether a given protein first got destabilized and then was positively selected for a change that stabilized it again. Okay? And there is a lot of positive selection, much more than we maybe thought 10 years ago, but much of that is actually not very species specific. So especially, say, in the immune system, you have a lot of evidence for positive selection because they interact with bacteria and viruses. And these things change all the time, right? But this is positive selection also maybe on the human lineage, but that's not very specific to the human niche, right? So that's something that is positively selected all over again, right? So to what extent positive selection is species specific versus more widespread across mammals or primates is an issue that has not been. And what doesn't go away is that even if you infer selection reliably for a particular locus, it doesn't tell you anything about function, right? I mean, which function was selected, you need to get from some other data. All right. So that's how it looks like, right? We have the catalog of changes. We have various ways which help, but of course do not totally solve the question which of them have been functional. And if you have plausible evidence from these three areas, and plausible is of course a pretty vague finding, but that's how it is, we might come up with a particular hypothesis that particular changes actually are linked to a particular phenotype. What is totally clear is that if I do that, first of all, the information is usually not enough to be really sure or to be really plausible. In some cases it is pretty plausible, in others it's very vague, right, as it is. But even if I have a very plausible hypothesis, I still want to understand more about the mechanism. So if I make this hypothesis, I want to go on, right? So I need to take the next step to somehow compare then two alleles, namely the derived human allele of a gene or a genetic element, and the ancestral or the chimpanzee version or the mouse version, depends what is a good proxy for the ancestral version, to a gene X in some model system, right? That's just the usual experimental approach. And I cannot take humans, I cannot take chimps, actually I cannot also take other primates for financial, ethical, practical reasons. I'm bound to use model systems that normally biologists use, biochemistry, cell culture, and the mouse. I come back to that. I think that is the major bottleneck to understand functionally human evolution. If you come up with clever assays that are valid, right, that really tell you something about the phenotype you're interested in, then you gain a lot. Say, for example, opsins, right? I mean, these small proteins that are in our retina and absorb light. For opsins, it's pretty nice. There's a pretty good correlation between a biochemical assay that measures their absorption and the phenotypic trait that tells you which colors the species sees, right? So in this case, you have a pretty good model system to infer which colors are seen by a given species using a biochemical assay. That's because we understand opsins quite well, right? In many other cases, right? And especially about the case we're talking today, that's of course very difficult. Very often, the things we're interested in are actually not working well in isolation, but actually have to do with a complex organic system, the brain, or also other systems. So, yeah. Anyway, so one way, and that's only one way how it could go about that then, is you start with a phenotype of interest. That's kind of how you would start with a certain disease and look for candidate genes, right? So that would be a candidate approach somehow. And as I said, there can be much, the knowledge about the human phenotypes could be much better, but for some of them we know something, right? AIDS susceptibility is something that that is different between humans and chimpanzees, some other diseases. We have a double maximum lifespan, which is pretty interesting. Yeah, famously we have a larger brain, and of course, language being, obviously there's a there's a phenotypic change on the human lineage. That's not to be discussed, but that's of course a very complex issue. So maybe one could start with speech, because that's clearly very different between humans and chimps. So, of course, while primates use sounds to communicate, I actually would have hoped that Tecumseh would allude on that much more, but now I have to do that myself. So while primates use sounds to communicate, like the vervet monkeys we'll also hear something about, I think, they essentially have an innate vocal repertoire, right? So it's not a voluntary movement. And also Constanze will probably explain much better than I can do that what vocal learning is. And historically, and also with chimps and with other primates, people have tried to teach them vocalizations. And that was interesting. That's just an anecdote, but I talked to a researcher that tried to train rhesus monkeys to make vocalizations voluntarily and give them rewards. And he said it was really mean, because you could see that they really wanted to make that, but they were just not able to do that, right? I mean, they just have a very poor voluntary control over their vocal apparatus. And that seems to be the case for essentially most primates. And there is a myth out there that would be anatomically based, that the position of our larynx allows us to vocalize voluntarily. Although it might be that the lower larynx in humans is an adaptation to speech, it's clearly not a reason why they cannot speak, right? So it has some neurological basis. So, yeah. And interestingly, as you know, right, I mean, if you teach chimpanzees or bonobos sign languages, they can pick up much more words and also partly combine them, but yeah. So whereas it's kind of not really clear if we have a higher cancer susceptibility than our ancestors, it's pretty clear that we have changed somewhere in the phenotype that has to do with vocal learning and speech, quantitatively and qualitatively. And it's very likely that this has a genetic basis, right? I mean, there are, of course, no real proof. There are no twin studies in that case. But yeah, we're...
But wouldn't twin studies be showing something different anyway? They would be showing about genetic variation between...
One would need to make crosses between humans and chimps to really tell. That's the problem.
No, I just feel that if you say there's a genetic basis, there's a big difference between saying that all humans have got a particular gene that does something.
The genetic basis is referring to fixed genetic changes, really. And this is variation that is not accessible anymore, which gives me the problem that I cannot use human variation really to make genotype-phenotype associations. So at that point, it's basically an assumption that there were genetic changes that tuned the human brain to make vocal learning, meaning speech, possible, which is, I think, not a very bold assumption in the end. Vocal learning does exist, as we will hear much more. It is rather rare among mammals, for not really clear, but apparently not many mammals need to learn vocalizations. That's just how it is. But it's, of course, not unique to humans. And especially songbirds are as we hear tomorrow, are a very well-studied system where you can understand how vocal learning appears. So this comparative approach here for speech has the advantage that you have other lineages during mammalian and bird evolution that actually also made this change, which is of course helpful to acquire additional information, as we'll see tomorrow. Okay, here we go. How could we do that? We start with gene function, and yeah, here comes in the gene that we have heard so much about. So with the KE family and with the mapping of the causative mutation by Simon, FOXP2 was kind of the first gene that was clearly linked somehow to this phenotype. And when I gave this talk, now more than 10 years ago, I thought, but there will be surely more, right? And there are much less than than I would have hoped, right? I think we would do that approach with a couple of more genes. But actually, yeah, we still talk so much about FOXP2 because, I mean, there are other genes like catnap, but the strengths and also the quality of the phenotypic investigation of that gene is just much better than anything else. Okay, so as Simon already pointed out, there are different types of mutations that have been found, including this mutation in the KE family. And essentially what is important, all these alleles are null alleles, right? They destroy the function of the protein one way or the other. Which then comes down to that if you have two normal copies, you have normal speech and language development. If you have just one functional copy, you have these speech and language difficulties that have been described to you by Simon and Kate and that you know. So that's the genetic situation, okay? When I started investigating that during my PhD, there were no genomes available, only for the human and only partially for the mouse. So we looked at the mouse sequence and we were pretty disappointed because it was so freaking similar between humans and mice. So among these 715 amino acids, there were just 3 differences. And actually that's, if you compare that to other human mouse proteins, that belongs to the 5% most conserved proteins you have if you compare mouse and humans. But surprisingly, kind of, the chimp has just one difference, so by inference, these 2 changes must have happened after we split from the chimps. Making that, given this long period of time having only 1 amino acid change, and in this short period of time having 2, making this more than you expect. This is still a pretty, I mean, if you now would go and say, okay, but we have 20,000 genes, I need to correct for multiple testing, you're out, right? Clearly. That's what I mean, that we have limited information with these positive selection signals. But still, right? And if you look actually across primates, this pattern is true. And now where we have more genomes, given that these 0.6% nucleotide substitutions is not very long, these 2 amino acid changes in FOXP2 is more than you get for all other lineages you have. There are some lineages that have more changes, like here bats, and it's not really clear why that is. Unfortunately, we looked a long time ago already with Constanze whether changes in the protein on the lineages that evolved vocal learning in the birds would actually go together with, would correlate with FOXP2 changes, but that's not the case. But that would be maybe too good. All right, so what remains is you have these 2 amino acid changes, which are more than you would expect for such a conserved protein. You have the phenotypic change of vocal learning that is pretty clear, and you have the gene functional link between coming from the KE family, which results in this hypothesis, right? Which is pretty famous. So essentially, these 2 amino acid changes are thought to somehow contribute to the evolution of speech and language. There's a side story to that is when did these changes happen? So initially we didn't have the Neanderthal genome sequence, but then in 2007 Johannes looked specifically at these positions, and now we have more genome sequences from ancient humans. And it's totally clear that all humans have these 2 amino acid changes, right? They're fixed worldwide, so that's good. But they also are shared by Neanderthals and Denisovans, meaning that they arose before humans and Neanderthals split. If we now would know exactly how Neanderthals spoke or used language, then that would be actually informative. I think we have no clue, really no clue, how much they might have spoken. So it doesn't really help us to further nail down the phenotype. Yeah?
It was interesting when the Neanderthal result came out, and I got some phone calls from journalists, and I was going, oh no, the journalists are going to go, this show proves that Neanderthals could speak, and you still see this kind of,
Yeah.
Interestingly, you're saying, well, we don't know about the Neanderthals, so we can't infer information about FOXP2, whereas in many people's mind, they use the FOXP2 results to make a prediction about whether Neanderthals could speak. So I just wonder what your...
I mean, you can view it statistically, and it depends on your prior, right? If your prior is very strong towards that these 2 amino acid changes influence speech and language, then this makes strong inference about what Neanderthals can do. If you are more careful about your priors, then this gives less information. I would be on the more careful side. Interestingly, and that's for me, that's really mostly I usually do not talk about this. So in the initial paper, we actually saw a signal close to these 2 amino acid changes that looked like a selective sweep, right? These regions that are very young and across the genomes. And initially, we thought that that would actually could have been caused by one of these 2 amino acid changes and strengthens the story. But later on, we found out, first of all, that if the Neanderthals had that, it's very unlikely that that could have been a sweep, right? Because that would push back the fixation of these changes so much that the sweep signal is unlikely to be caused by these things, right? And there are other arguments why the amino acid changes cannot cause the selective sweep signal. And then a recent study, Tommy and Svante's lab, looked at Neanderthals and looked basically at all the positions in that region that happened on this lineage, and he found one in a regulatory element that happened here on this lineage and could potentially be the cause of the sweep. So that would be a different change in FOXP2. I think the prior that these amino acid changes are functional is better than the sweep signal, because I actually think that what we know about the signal now, it gets less likely that it's really a sweep. And also the regulatory element is not really clear what it does. It affects the strength of a transcription factor binding, but we do not really know in which tissue it's expressed, because the mouse doesn't express this region. So it's very unclear what that does. So I will focus on these two, because we have not looked much more on this additional change. But I would be more skeptical that it's really functional, actually. Okay. So, as I said before, we now have this hypothesis. We want to compare the human version of FOXP2 to the ancestral or the chimpanzee version of FOXP2, which is in this case very identical, and have an assay system where we can compare phenotypes that are relevant, right, to this question. And that's, of course, and as I said, right, we cannot cross or back-cross humans and chimps. We cannot genetically manipulate them. We could find back mutations. I mean, we're so many humans, right, that some of humans run out there with a back mutation to the chimp state. But yes, I'll take you with me. No. And eventually it might actually be the case that if everybody gets sequenced this genome, that you find these people, but of course they're rare, right? I mean, you can calculate from the mutation rate that maybe each generation you have 200 chromosomes born worldwide that carry a particular back mutation. So it's not really a project. And biochemistry is nice, but of course does not really scale to the question we have, to what extent that might have to do with speech and language. Also, cell lines, we do not have really good assays on a cellular level. But we have the mouse, and that's really the only option. We have to study such a change in a developing organism. So that's what we did. Nicely, these 2 amino acid changes are close together, so we can make a very careful genetic manipulation of the mouse by knocking in the normal FOXP2 gene of the mouse these 2 amino acid changes, right? So we just change these 2 amino acid changes, which our hypothesis is about. And then we can cross these mice and actually also get rid of this neomycin cassette if you're really careful what we did. And then you can compare littermates that are homozygous for this humanized version of FOXP2 to their normal wild-type littermates that carry the chimp or the mouse version of FOXP2, right? And then we can directly compare the effect of these 2 amino acids in a mouse model. This is not the same as a mouse model that has 1 or 2 copies deleted, right, of FOXP2. That's a totally different allele. That's a related but very different question. So, but they're also important because these mice somehow are the model for the KE family. So if you want to look for phenotypes that are informative for the speech impairment or for the speech evolution, then it certainly helps to know what happens with KE mice, right? That's pretty clear. And yeah, we have these mice now for a long time. I have no sound, but they talk, of course. Right. No, don't have the sound file, sorry. No, they don't talk, obviously.
What's the difference between humanized and wild type?
In talking?
No, I just know something very basic about the difference.
Maybe again? So,
They're not fully humanized.
so
Just
they have, in the normal FOXP2 gene of the mouse, they have changed these 2 amino acid changes that have changed on the human lineage. So the whole regulation, that it's a normal FOXP2 gene of the mouse, but has these 2 amino acid changes, which is our hypothesis about, right? That's as good as it gets to build it on the genetic side to build a model. So so what comes back, right? If FOXP2 would be very restrictive in the location where it's expressed, right? If it would be only expressed in Broca's area, that would constrain our ideas pretty much. But in fact, it's actually expressed in essentially all tissues people have looked at, and then interestingly within tissues in a subset of the cell types. So these are typical patterns, which is interesting because, I mean, it's not in every cell, but it's in a subset of cells which would go together that it contributes to specifying cell type identity in many different organs. So in the sense, it's also rather typical because, yeah, it's expressed in many organs, but not in all cell types. So I think that you would find several transcription factors that would have maybe a pattern like that. And also within the brain, it's not expressed in all, it's expressed in neurons, as already discussed in layer 6 and a bit in layer 5, and especially in the striatum, as you've heard by Sonia, and in the Purkinje cells of the cerebellum. So there's some specificity, but it's not kind of overwhelming. Interesting, the hippocampus has really no FOXP2 expression by all what has been seen so far. So that constrains maybe some functional consequences. And maybe one question one could ask is, which of these different organ systems where it's expressed is now affected if I humanize my FOXP2? And could such changes be related to the evolution of speech and language, right? And importantly, what we did is we screened these mice in the so-called German mouse clinic. And clinic is really a mean term because it's not about healing mice, actually. But it's a clinic in the sense that they have set up these screens, right? So you give them 15 mice per genotype and sex, and they hop through these different labs, which all have their screens set up that test behavior and immunology and metabolism, etc. And these humanized FOXP2 mice, compared to their wild-type littermates, are very, very healthy. So they're totally doing fine, which is also nice for them and nice for me because they don't suffer really from having the human version of FOXP2. And they show no difference in all these 250 parameters that were assessed here, except that they explore the environment in this behavioral assay slightly more carefully, right? They stick a little bit more to the wall, do not look in the holes that much, etc. So that's the only repeatable phenotype that we found. The knockout mice, interestingly, so the heterozygous knockouts, right? The homozygous knockout mice are dead. We discussed that already with Sonia. So I think dead mice are medium informative for the speech phenotype. So I think these are really the mice that matter most. Interestingly, they're also totally healthy. They're totally fine. And they show a variety of very subtle but significant differences across a range of these traits, right? So you find slight alterations in metabolism and lung function and blood pressure and also hearing. So probably reflecting that although also the mouse does fine with just one copy of FOXP2, they're healthy, they run around there. But if you look carefully enough, you see slight effects in these different tissues where FOXP2 is expressed.
Can
And
I just ask,
huh?
what's the genetic background against which this is all happening? When you say wild type, I mean, are they a particular inbred strain? Or?
Yeah, I mean, you start with, yeah, it's all black 6 background from the beginning. So unless you have a very tight linkage of the cell line where you created the knock-in that explains that, then it would be a strain effect. But that would be very unlikely given that the cell where the mutation was introduced is black 6 and then was back crossed to black 6 now more than...
You're
okay.
completely talking over my head, but I just sort of wondered why you wouldn't just get a very heterogeneous group of mice to look at this in.
No, I mean, you really compared directly randomized to their wild-type litter mates, right? They're sisters and brothers
Yes.
that run through this clinic.
Yes.
So you have you have a control that
Yes.
doesn't
No,
get
I
any
think
better.
the only reason I was asking was I just wondered whether the impact might depend on what the genotype was elsewhere.
That is something you usually find, that if you would now transfer this to a different mouse genotype, it's not really clear if you would find the same things again.
Yeah, that's something which we didn't really bring out in our earlier talks, but in general in the mouse behavior, well, phenotyping field, that backgrounds can make differences to the kinds of effects that you see, and it's a problem that, of course, the amount of work it takes to do this, you can't, but they are trying to, on some of the autism genes that people are studying, they're now trying to study them on multiple different backgrounds, so that
Yeah.
clear idea.
I mean, in generally, you study these things in a particular environment, in a particular genomic context, right? The biggest problem is that it's a mouse, I think.
You know,
And it also depends how you interpret that, right? I mean, a gene-gene interaction also means that the system is affected. So it's a question how generalizable that particular phenotype is, right? If you maybe have more T cells for this given phenotype, does it mean that it somehow has to do with the immune system or that it specifically has to do with T cells, right? So depending on how specific your interpretation is, it also then matters what the background effect, right? So I think the interpretation here is that, so the main thing that I got out of there is, first of all, the heterozygous knockouts show subtle effects in a variety of screens. And for the one behavioral screen, they tend to show the opposite phenotype from the humanized mice. Yeah?
I'm interested in that phenotype. What does it mean? You said they were
That's a good question.
before. Does that mean they took
I come back to that a bit later, but I have to speed up, I guess, huh? Not much later, no. Okay, I go through that a bit more quicker. So we threw really over the last 3 years many tests of that. One thing we also did, we measured dopamine and also serotonin levels. And if you measure tissue dopamine content, it is reduced in the humanized mice and it's increased in the heterozygous knockout mice. Interestingly, and that's new, not yet published data, we also did mice that do not have the 2 amino acid changes knocked in, but 2 separate lines that have one or the other mutation knocked in. And I think that's a very important finding here, that you essentially get a reduction in dopamine levels for both of these amino acid changes separately. And that's pretty important because it not only means that you have 2 amino acid changes that are more than you expect, but for this phenotype, they push in the same direction, which is actually the hallmark of directional selection. If the one would have pushed that in this direction and the other in this, I would have been extremely worried. But for this phenotype, we have not really looked so closely at others. It seems like it's clear that they both reduce dopamine levels and actually also serotonin levels in a very region-specific manner. But importantly, they both do that. Maybe even additively, but we do not have really enough power.
So what does this mean, though, for trying to understand which of these amino acids is the functional chain? Or are you saying that that was the idea of generating these two lines, was to say... Okay,
actually, that was the initial idea, but now I like this much better.
Okay.
Because it really shows that both of them were probably... I mean, if this is a relevant phenotype for the thing that happened during human evolution, which I increasingly think that that might be actually a very relevant phenotype, then that means that both of these changes pushed it in the same direction, right? You have twice a change that goes into the same direction. And that's also statistically not terrible much more, but it's much better than if they would have gone in opposite direction.
But some of the other findings, electrophysiology, for instance, is also both the same direction?
So that's something where I disagree with Svante. So the stronger effect in electrophysiology is seen for this change. From a statistical point of view, I say that we cannot really say at this moment whether this has no effect. It depends a bit on the null, right? So if anything, this has a stronger effect. For the dopamine effect, it is very clear that both of them do that. In my opinion, for the electrophysiology, we have not enough power. In Svante's view, it doesn't do anything. Oh, I will never end. Time. Oh yeah, wait. Okay, I try to. So, dopamine links to corticostriatal or the cortical-basal ganglia circuits. Dopaminergic cells are mainly here. They project especially to the striatum, where the input from the cerebral cortex gets integrated with this dopaminergic signal, which is a reward prediction arrow. So basically, if you do something and you get a reward without expecting it, your dopaminergic neurons fire. And that helps you to learn which actions are actually favorable. That's a very, very shortcut version of what this does. And as you have already heard from Sonia, right, in these medium spiny neurons, FOXP2 is expressed really a lot. So the dopamine phenotype plus also the exploratory phenotype leads a bit to these regions. And as you know, the striatum is something that is affected in the KE family. So the part of the striatum is called caudate nucleus in humans, and that has less gray matter. And as you hear tomorrow much more, the specialized vocal learning area of songbirds, if you reduce FOXP2 levels there, you actually also impair vocal imitation. So there are a few reasons, in addition to the dopamine finding, that this might be a site of action. We looked at many different things, some by chance, some more targeted. We see a difference in neural outgrowths in these neural progenitors that we isolate from these mice. With this assay, that assay was more variable than I wished, and I have given up on that, but that may be just my... the postdoc that worked on that was not able to produce that stably. But anyway, so, and in vivo, actually, if you stain them with Golgi, you see also this effect that the striatal neurons have longer neurites. We measured striatal synaptic plasticity in these neurons. So you patch these cells, and a very common assay you do is you apply a high-frequency stimulus to these cells, and after this high-frequency stimulus, which was already a bit explained by Sonia yesterday, they react less than before, right? So this is a form of synaptic plasticity called long-term depression, which is in these cells very frequently studied. And if you do that, and that was initially done by Johannes Schwarz at the neurology department in Leipzig, we see that the humanized mice show a stronger reaction than the wild-type mice. Again, that's opposite to the findings for the heterozygous knockout mice, or in this case, the KE mice by Matthias Grosser, which showed kind of that these mice do show less or even no reaction to the same protocol. We have worked a bit on the mechanism, and that's also actually new. So we seem to have lucky because our protocol was a rather mild protocol. If you apply a stronger protocol that also depolarizes the cells during the high-frequency stimulation, you actually see no difference. So it seems that with a mild protocol, the cells are more ready to go to LTD than the wild type cells. But of course, if they're both at the wall, right, if you both push them totally, then they end up both at the wall. So that explains maybe a bit about the mechanism, and then we also see that the stronger LTD in these humanized mice depends on D2 receptors and on postsynaptic NMDA receptors. So if you inhibit inside the cell the NMDA receptors, you get a weaker, you get no difference between the wild type and the humanized anymore. So we think that although the number of NMDA receptors seems unaltered, we see that this electrophysiological phenotype depends on NMDA and D2 receptors.
If you look at the ratio, NMDA,
Yeah, it's not changed. No. Yeah, we basically do not know currently what it is, but of course there could be many reasons, modifications in the NMDA receptors that will alter that. Anything else that would leave in more calcium. There are many possibilities, of course. We also looked a bit at the specificity of the effect using this neuronal phenotype of the longer dendrites. So while in the striatum you see that I showed before, right? You have longer dendrites in the humanized mice. You do not find that in regions where you do not have FOXP2 expressed, but you find it in regions of layer 6 of the striatum and the thalamus, and you do not find it in Purkinje cells in the amygdala. So if you would go that the Purkinje cells in the amygdala are not parts really of the cortical basal ganglia circuits, we see an effect in cortex, in the striatum and the thalamus, but we do not see anything in the Purkinje cells. So we think that the humanized effect rather affects the cortical basal ganglia circuits than the Purkinje cells. We also looked at synaptic plasticity in Purkinje cells, and this is actually unchanged by many different measures that Wolf did. So currently the idea is that this humanized FOXP2 especially affects cortical basal ganglia circuits. We initially thought that dopaminergic neurons do not contain FOXP2. That's true for adults, but actually it seems to be the case that during development FOXP2 is expressed in dopaminergic cells, so that's actually a site which could also be causative for these findings. Okay. So we think we have really shown that the humanized version alters properties of these cortical basal ganglia circuits, which on the one side seems not very big effects, but then on the other side, if I think about these are just 2 amino acid changes, once in a while I'm really stunned by the magnitude of the effects and how lucky I was. And what is also important, I think, that at least for a good subset of these phenotypes, genetically you can really talk about a gain of function. So it is the opposite direction of the effects that you see in the heterozygous knockout mice. And that, I think, in addition to these 2 single amino acid changes, is a pretty strong argument that that it is functionally relevant, at least to some extent. Because if it would be the same direction as the heterozygous knockouts, I would rather interpret that as the mouse doesn't really work well together with the human FOXP2. But since it's the opposite phenotype, you could genetically talk about a gain of function. That still doesn't help us much to understand speech at that point. So the question is, can we find complex phenotypes that are more informative? I already mentioned that in this one screen, right? They explored the environment more. So yeah, these are the heterozygous knockouts, and these are the humanized mice. So you have a significant reduction in the way how they explore the environment. But the problem is here that you assay all different kinds of behaviors, and it's not really clear how to go from there. We also, and we discussed it already a bit, scored vocalizations. So pub vocalizations, that was the last experiment I did in the lab, I think. And what we found that these pups basically vocalize at a slightly lower, but significantly lower frequency. The problem here is that, first of all, the adults do not show this difference anymore. And second, it's pretty hard to go from there. We have no clue what would follow from that. It's clear that these vocalizations are not learned at all. And all different kinds of small anatomical changes that are very on the periphery of these effects could actually cause that. So I have no idea, although initially I was very excited, and I think Svante goes still around, that they vocalize differently, and that's what you read, because it's kind of obvious. And it could still be interesting, but I just don't know what to do with that. I think it doesn't, unfortunately, it doesn't help much, because too little is known about how this is wired, what could cause that, etc. So, yeah. A more recent thing which I find interesting, that if you that was done with Matthias Grosser in Paris, he found that the KE mice, if you give them cocaine, they do not run as much as the wild-type littermates upon cocaine injection, whereas our humanized again show the opposite phenotype. So that somehow means, again, that the cortico-basal ganglia circuits, if you activate them with cocaine, because that's what you do, react differently and in opposite direction in these two mouse strains. And in contrast to the vocalization, that's a phenotype where it's much, much more known about that. So we're following up that a bit. And the best thing I can offer you is done by Christiane Schreiweis, who was a PhD student in my lab and that spent a huge amount of time in Anne Graybiel's lab at MIT, because Anne Graybiel has set up a system where they look at association learning in mice. This T-maze works like that you have the mouse that sits here and it either encounters a rough surface like that or a smooth surface, and then gets rewarded either on the left or the right side depending on the surface. So what happens is that, so the mice learn that, right, and start with 50% chance, right, and get better and better and better, and that has been a lot of work. A huge amount of work. It's a terrible essay, really. But what's very clear is that the humanized mice learn this association faster than the wild-type mice. So in this particular paradigm, the humanized mice do learn these associations faster. And it gets a bit more complicated because, more or less by chance, Christiane found out that if she takes away the spatial cues that happen to be present, if there are no spatial cues, the difference is gone. If she puts it back, you see it again. So I was really a long time worried about statistics here. But now I'm sure. So that's really a lot, because each day you have to do for each mouse 40 trials. So that's just a lot of time in the darkroom.
The humanized mice cannot do this without spatial cues?
There's no difference.
And you have
So essentially, the humanized mice do it better if spatial cues are also present.
Because this is what we see with a knockout fly, right? This is bizarre. We're going to talk about it tomorrow, but this is so bizarre.
Yeah, that's partly similar, I think. Yeah, I mean that.
Exactly like that. Without visual cue, they cannot do it, but with visual cue, they can.
Well, but it's not that they it's not that they can't do it. Right?
no
It's
difference.
that the humanized have an advantage.
They're faster.
But the grey
But
line
that goes away when they don't
It's
have
an
visual
effect
cue.
that goes away.
Yeah.
Yeah. So that might be consistent with.
the grey line in the second graph is as good as the black line in the first graph. So actually
can you
taking away
compare
the
across
cues
the graph?
makes everyone better.
So I mean, yeah.
Wild type better.
Yeah, it makes
But can you compare across?
So statistically, there are two ways, right? There is just the interaction between those two environments, and it's not really clear. But if you think you can compare them, then it's indeed the case that essentially the wild-type mice do worse in this condition. Statistically, I do not really care because it's a genotype-environment interaction. So it happens something to the genotype here. But maybe so Christiane continued from that.
But we know that the hippocampus and the striatum contribute to that kind of
Yeah.
navigation differently.
Yeah. So yeah, let's see how detailed that will get. So she tested other things. So actually, we also tested rotarod learning, which came up, and there was no difference. We always hoped that we would see an increased rotarod learning, but we didn't do that, and we really tested that. So Christiane tried to disentangle the spatial and procedural learning that goes on here together. And she made a task where she did a cross maze where the mice either learned on a response-based version, so they had to learn to turn left or to turn right, then switch to a place-based version, so they had to go to this side of the room, and then she switched back. And what she saw is no difference here, no difference here. So if you have these learning systems in isolation, you see no difference, and that's also compatible with, we saw no difference in other procedural tasks like the rotor-rod learning. But if they switch, which would be this transition from place-based to response-based learning, then they are faster. So since FOXP2 is, by all we know, not expressed in the hippocampus, which has been usually associated with spatial learning and the stratum associated with procedural learning, it has in recent years been clear that actually the different regions of the stratum contribute differently to these things. So it has been shown by several papers that spatial learning goes from the dorsomedial striatum, which is hooked up to the associated cortex, and the procedural learning is hooked up to the motor cortex and this dorsolateral striatum. So you have two involved striatal regions, dorsomedial, that is associated with place-based learning, and dorsolateral striatum, which is associated with procedural learning. And the idea is basically that this transition that happens in normal circumstances when you proceduralize something is easier for the humanized FOXP2 mice. That's currently our interpretation of the behavioral data.
Could it relate to this business about being more careful in exploring the environment? I was thinking particularly with the rough and smooth surfaces. I mean, if one strain is just more aware of the rough smooth difference, would they then not learn better using that cue? And they might be more aware if they're sort of rather careful.
So we have no really evidence that there are some sensory differences or that I mean, the link in my view would rather come from an alteration in the dopaminergic system that has effects on exploratory behavior and this type of learning. But eventually it's of course pretty hard to, as always, to distinguish cause and effect.
Got them evaluated for that, you could see if it was a predictor that could explain the difference.
Yeah, I mean, there's no indication from speed how they went through these mazes, and there's really no indication that would hint that it's actually a more cautious exploration that would lead to this phenotype. Plus, you would expect then also that you see that with the without the spatial cues as well, right? So that wouldn't explain that either. So
latency to enter the maze is the same?
Same.
I guess you'd pick up in other tests as well, testing anxiety and things like that. Right?
So no anxiety, I mean, we checked also anxiety tests and it's all not changed. So basically what these behavioral results led to is that maybe this balance between those two regions, right, that have to do with spatial and procedural learning somehow altered, and we were looking for additional hints that this might be the case. And the one thing we did, so that's actually one of my main expertise in the lab now, to do RNA-seq, and in this case we cut out by laser capture dissection part of the cortex and the dorsomedial striatum and the dorsolateral striatum of 14 females and 16 males, all 8 weeks old, so that's a reasonably powered study. And we saw actually no difference in the cortex, so we focused on the dorsomedial and dorsolateral striatum for this particular question. While you see a huge amount of differences between those two regions, which are pretty spatially close, right, which surprised me, which to some extent tells you that you really have a huge amount of power to see differences if they are there, we see no significant differences between the genotypes on a single gene level, which is a negative finding in some sense, but actually also a positive finding indicating there are no major cellular or structural rearrangements, right, that would explain the difference between those two genotypes. We have seen that partly before, and we've seen that also for the heterozygous knockout mice. There are no big differences in gene expression patterns. If we look at the level of gene grouping, so if you group these differences into functional categories, we see, and we do permutations tests, which I think are really important to the proper statistics. We see a very slight effect of genotype. So the distribution of FOXP2 downregulated genes is not random across functional groups, and the biggest differences we see between DMS and DLS have to do with neurotransmitter transporter activity, and these less interpretable groups. But so the take-home message there is some indication that DMS and DLS have slightly different effects. There are no major cellular differences, and if it might have to do with neurotransporter activity and these kind of groups. But it's more remarkable that there are so few differences, actually. I think that's the main result there. Electrophysiologically, it's interesting that this is the result we have seen before. Dorsolaterally, you have a stronger LTD. Dorsomedially, you tend to have the opposite effect, which is an interaction that is significant. And the dopamine is also interesting, and we see this consistently in all these single amino acid changes too. The dopamine levels are also regionally specific differently. So the dorsomedial stratum has lower dopamine levels, whereas the dorsolateral has actually no differences. So essence, we see on several levels an indication that dorsomedial and dorsolateral stratum is differently affected, which might relate to the behavioral findings. And then we can sum up that all together and speculate how that might relate. So that's currently as good as it gets on my side. We hope it gets better, of course, but how can we now speculate how that might relate to speech and language? So of course, I entered that because it's so obvious to me that I keep saying that of course, it's no reason to believe that that's the only change, right? That's totally crystal clear, right? That if at all, this is one piece of the puzzle. And how many there are, it's totally unclear, right? But just sure, right? So that's pretty certain. So I think that if it's really true that it affects cortical basal ganglia circuits, then given that these circuits seem not to play such a huge role in adult processing of speech or language, but more in the acquisition of language, that also the effects would have rather to do with acquisition than actual processing. That would be one speculation. What I find very interesting, and you will hear more about that, is that songbirds repeatedly have evolved a specialized cortical basal ganglia circuits, where the area X is kind of the striatal part of that, that has to do with vocal learning. So from this comparative study in birds, it seems clear that you specialize these circuits for vocal learning. And it might be a plausible explanation that that actually also happened when humans evolved vocal learning. So FOXP2 might be in this package that you get if you adapt your cortical basal ganglia circuits for vocal learning. And more directly, if you directly interpret the behavioral finding, it might be a fast transition, right, from declarative in mice spatial learning to procedural learning, and you could speculate that that might play a role in speech and language learning. Could also be more indirect effects that you observe in these mice. What I find interesting that if it affects in a larger sense these circuits and dopaminergic signaling maybe even, what you probably need if you want to vocally learn as a baby, you need to reward it, right? So you need to probably change the rewards maybe by wiring up the circuits to dopamine in a stronger way, because otherwise it's hard to imagine how these kids would actually babble so much, right? I mean, somehow they must find it rewarding to vocally imitate, and that must be somehow genetically. And yeah, tuning stochastic learning could play a role in a more indirect sense. Yeah, and overall, that is the data that we know, and that's more new now, that they learn the associations faster, that they have this altered balance of these two subcircuits, and potentially that could relate to speech and language evolution. And further, what we do not really understand, which is important, where and when do these changes actually happen, right? Is it during development? Is it acutely? Is it in the dopaminergic neurons? Does it happen in the striatal neurons? Is it maybe some other neurons in this network? It's just unclear, as usually, because the mouse is a complex thing, right? And we try to, I think we need to solve that before we can go more deep into which genes are regulated and affected, and how do these targets evolve. And we work on also a bit now on the biochemical mechanisms. But it's actually, I think it's very important to find the specific cell type and developmental stage where it matters before you spend much time on these things. I'm done. Sorry for long.
We're going to have a few minutes for questions, and then we're going to, because we're running a bit late, we have a barbecue that's from 6 till 7, but we thought that actually instead of starting the debate at 7, we would start at 8. Is that okay with people? So it gives us more time to eat at the barbecue.
The barbecue is in the pool or at the pool?
In the pool. With your, yeah, bathing suit. I don't know. I guess it's by the pool. I don't know. But anyway, shall we take some? If any guys have got any questions?
I just had a question about the behavioural phenotypes of the. You were saying about the functional gains of the humanized mice, and then you had on your list of the knockout mice that they had some subtle difference in their hearing. I just wondered whether you could say a bit about how you test mice hearing, what the differences were.
So this test was in this mouse clinic, and they do clicks. Sorry? So how we assessed, so we said the heterozygous knockout mice hear less well, and how we assess that. That's a very simple test. You click, and you have a clicker, and you look at the reactions.
I was just wondering about the measures and whether the sensitivity is the same, whether there's a gain or a loss, and whether if the humanized ones had some kind of sensory gain, whether you'd be able to figure out.
No, so the heterozygous knockout mice have a slight defect in hearing.
Yeah.
The humanized mice are totally the same as the wild type littermates.
But is it the kind of test where if they were, are they able to show them to be better?
Yeah. I mean, not all of the tests can actually show a gain of function, but this one can. Yeah.
What do we know about FOXP1, given like all the genetic redundancy that we know about, and how could you test this in the humanized version? How?
So we know that FOXP1 has no change on the human lineage.
Okay.
That's kind of good, I think, because it makes my life much easier. But of course, right, this is just coming back to the genomic annotation. This is only protein. We do not know where the regulatory reasons for FOXP1 are in the relevant types, and which of these changes might actually change the expression of FOXP1, so we're blind to this part. And yeah, I mean...
So you can't do very much about it?
No.
So why do you think there are 0 differentially expressed genes between the genotypes?
Well, 0 is a... Actually, I think there is not 0, but if you look at the single level, despite that we really have high power with 12 animals per genotype, and you can see the high power that you see really a lot of differences between the two regions. Let's put it like that. I think you have very often two stages, also in all terms of types of analysis that do knockout mice or also cell lines. You either have a stable system, right? So you do not change it, and then you have very hard times to see differences. I think then often people cheat statistically, to be honest. Or you have a really strong effect on the system, and then you have huge amount of differences, right? I've seen many data sets that are either of these two, and I think I think that could be. But on a very subtle level, we see this evidence that on a group level we see slight changes in gene expression. And that could be because if only a small subset of the cells of the sample show actually the difference. For example, if FOXP2 is acutely involved in the cascade that integrates dopaminergic and glutamatergic signaling, then only a few cells would actually be active, say 1%. I don't know how many cells would be in that state if you take a mouse out of the cage. And then, of course, you would, if there are a small set of genes differently expressed in only 1% of the cells, then you do not see much anymore. So either it's not the right place or time to see differences, or it's not the right environmental conditions. So we try to now look at mice that have cocaine-induced acutely and then push more cells into the state and then see if we see difference.
But we're not going to give cocaine to K family, right?
You're not?
Maybe.
They use it already.
Maybe we could.
I mean, it isn't a lot
Do you
more
think-
one would.
Do you think we could do that?
For certain questions, I don't think we're allowed to answer. Gina taught me to say that.
Question over here.
Have you looked at the differences in the social behavior, like social recognition, memory, social learning?
Not so terribly much, so a bit that's assessed in this modified whole board, because they also can, you can measure the time they spend with their cage mates. But except that they walk around, that they explore the environment less, we did not see any social interaction differences. And a few of them have been done also by Matthias in this context, and we have not seen any of them. I'm not quite sure if I would expect really any. Yeah, but hard to say. No, so far we have not found any.
Okay, last question from Kate.
Well, so I was just thinking about
We had a
this.
student here.
Oh, sorry, I didn't see the student. Sorry. Last question from Tulia.
I was just wondering if there's any intracellular differences in intracellular localization of the wild type or human virus.
We have looked a bit, but no, not so far. I think that would be. I think one would need to measure much more carefully. That's the thing, right? If you destroy the function, right, and it's like Sonia showed outside of the nucleus, then it's a pretty strong allele, right? But here you really fine-tune. I mean, the FOXP2 still needs to work, right? So you just fine-tune FOXP2 functions. So I actually do not expect to see big effects, because that would be also unlikely evolutionarily, right? Evolutionary changes do usually not get by with big effects, especially for these pleiotropic genes that have so many different things to do. So I would not expect any such a drastic phenotype.
Okay, I think, Kate, you're going to have to ask a question here with barbecue. And we'll be back. So we go to barbecue, and then back here at 8 o'clock. Right here.